98% of your DNA doesn't code for proteins.
Once dismissed as junk DNA, these mysterious regions may contain extraterrestrial coding patterns. Recent research from St. Jude Hospital discovered over 500 functional non-coding DNA variants influencing medical treatment responses, suggesting deeper significance we'll explore later. While mainstream science pursues natural evolution theories, a puzzling historical gap exists. Around 100,000 years ago, human intelligence dramatically accelerated, separating us from every other species on Earth. What triggered this cognitive explosion? Could an external intelligence have programmed these non-coding regions? The answer might be hidden within these enigmatic stretches of our genome that scientists are only beginning to understand.
The mystery of non-coding DNA. What if the majority of your genetic code isn't actually yours? When scientists first mapped the human genome, they encountered a profound mystery. Only about 2% of our DNA actually codes for proteins, the building blocks that make you who you are physically. The remaining 98% appeared to have no obvious function. This vast genetic territory was quickly labeled junk DNA, dismissed as evolutionary leftovers, genetic debris accumulated through millions of years of evolution. The term junk DNA gained popularity in the 1970s, and for decades, these non-coding regions were considered useless genetic baggage. Scientists focused almost exclusively on the protein coding regions, believing they held all the significant information worth studying. The rest was viewed as genetic noise, silent and meaningless. This dismissal contradicts a basic principle of evolution. Nature is remarkably efficient, rarely maintaining useless features across millennia. Natural selection eliminates waste. So why would humans preserve such enormous quantities of supposedly useless genetic material? The metabolic cost of replicating this DNA with each cell division is significant. From an evolutionary perspective, carrying genetic deadweight makes little sense. The scientific tide began to turn in the early 2000s. Like opening a long-overlooked treasure chest, researchers began discovering valuable contents within this junk. Dr. Ian Konigsberg and his team at the Cambridge Institute for Genomic Research found that these supposedly useless regions serve crucial functions in determining the three-dimensional architecture of DNA within the nucleus, essential for proper gene expression. These non-coding regions contain regulatory elements that act like dimmer switches, turning genes up or down depending on the cell's needs. They influence when, where, and how much protein gets produced. Without these regulators, our cells would be like factories with machines but no operators controlling them. St. Jude Children's Research Hospital researchers amplified this significance through clinical applications. They discovered that these regions contain instructions affecting patient responses to medical therapies. Their team identified over 500 functional non-coding DNA variants associated with chemotherapy resistance in childhood leukemia. These variants don't change proteins directly but alter how genes respond to treatment. Two patients with seemingly identical leukemia can experience dramatically different outcomes based solely on differences in their non-coding DNA, a crucial insight for personalized medicine. Scientists now estimate that at least 80% of the genome is biochemically active. The ENCODE, Encyclopedia of DNA Elements, project has mapped thousands of regulatory elements throughout the non-coding genome, though this represents just the beginning of our understanding. Large portions remain mysterious. Some segments show functions unlike anything in other species, with patterns that don't match evolutionary expectations. These unexplained segments contain sequences that some researchers argue appear algorithmic rather than random, showing signs of complex, deliberate structure. Dr. Elena Makova at Pennsylvania State University has studied these unusual patterns for over a decade. Her analysis reveals mathematical relationships that appear designed. These sequences show organization that doesn't align with current models of genetic evolution, she explains. They contain patterns that more closely resemble engineered code than randomly mutated DNA. What's puzzling about these regulatory sequences is their apparent lack of evolutionary origin. While protein-coding genes typically have predecessors in other species, many regulatory elements in non-coding DNA appear to have emerged suddenly in the human genome, without clear evolutionary precursors. These genetic switches control crucial aspects of human development and cognition. They help determine brain structure, influence neural connections, and regulate processes associated with higher thinking. For example, specific enhancer elements in chromosome 8 activate genes critical for neuron migration during brain development, a process fundamental to human intelligence.
Another regulatory region on chromosome 16 controls FOXP2 expression timing, a gene essential for language acquisition. The concentration of unexplained regulatory elements around cognition-related genes has led some researchers to question their origin. Mainstream scientists offer alternative explanations. Perhaps these elements evolved through complex processes we don't yet understand, or emerged through viral insertions later repurposed by natural selection. These explanations, while scientifically plausible, still struggle to account for the specificity of the regulatory systems observed. As genetic sequencing technology advances, we continue discovering new functions for non-coding DNA. These regions contain essential instructions that make us uniquely human. They control aspects of our biology that separate us from other species, our cognitive abilities, language capacity, and extraordinary adaptability. The mystery of non-coding DNA continues unfolding. What was once dismissed as genetic garbage now appears to contain some of our most important biological instructions. These regulatory sequences with unclear evolutionary origins function like sophisticated genetic switches, controlling the development and operation of the human brain. As we examine these genetic mysteries more closely, we must ask, if these sequences aren't random junk, what purpose might they serve in our evolutionary story? The answer may fundamentally change our understanding of human origins and the forces that shaped our remarkable cognitive abilities. The Great Cognitive Leap, around 100,000 years ago, something extraordinary happened to our species. Something that science still struggles to explain through conventional evolutionary theory. This event fundamentally transformed human potential and set us on a path that would eventually lead to civilization, technology, and ultimately, our current global dominance. The transformation was cognitive, and its impact was profound. To appreciate the magnitude of this shift, we need to understand what came before. For millions of years, hominid evolution followed a predictable pattern. Our ancestors' cognitive abilities evolved at a slow, steady pace consistent with natural selection's gradual process. The fossil record reveals this progression clearly. Australopithecus, 400-500 cubic centimeters, Homo habilis, 600-700 cc, and Homo erectus, 800-1100 cc, a gradual increase spanning approximately 2 million years. Their technologies mirrored these cognitive capabilities. Early stone tools remained remarkably static. Oldowan choppers, 2.6 million years ago, and Acheulean hand axes, 1.7 million years ago, persisted with minimal innovation for hundreds of thousands of years, reflecting the steady but unhurried cognitive development of their creators. Then, like a dormant volcano suddenly erupting, human intelligence exploded. Around 100,000 years ago, archaeological evidence shows a dramatic emergence of behaviors indicating enhanced cognitive capabilities, abstract thinking, innovation, complex planning, and symbolic representation. The story of Blombos Cave in South Africa illustrates this transformation vividly. Archaeologists unearthed an ochre processing workshop dating back 100,000 years, complete with abalone shells serving as paint containers. Imagine early humans gathering around these shells, mixing red pigments for symbolic purposes, communicating through color and pattern in ways their ancestors never could. The site contains engraved ochre pieces showing geometric patterns that suggest abstract thought and early artistic expression. What makes this cognitive leap particularly puzzling is the absence of any clear environmental pressure that would drive such rapid advancement. The fossil record shows no sudden climate shift, predator threat, or resource change that would explain this intellectual growth spurt. This cognitive explosion manifested across multiple domains simultaneously. We see technological leaps with composite tools, spears with stone tips attached using complex adhesives, personal ornaments and decorated objects appear, demonstrating symbolic thinking. Archaeological sites reveal complex social structures and trade networks spanning hundreds of kilometers. Scientific views have evolved too. The traditional perspective once placed a cognitive revolution around 40,000 years ago, coinciding with European cave paintings. However, discoveries from Africa like the 77,000-year-old cross-hatched drawings from Blombos Cave prove that modern cognition emerged much earlier. This acceleration created an unprecedented gap between humans and our closest relatives. Despite sharing over 98% of our DNA with chimpanzees, our cognitive abilities differ fundamentally.
Chimpanzees display problem-solving abilities and can use simple tools, but lack capacity for abstract thought and complex language that characterizes all human societies. The evolutionary timeline proves problematic because it contradicts the gradual pace observed in other species' development. Complex traits typically develop through small, incremental changes over many generations. Yet with human cognition, the intermediate steps appear missing. We see steady, slow development for millions of years, then a sudden acceleration producing behaviorally modern humans in an evolutionary blink. The archaeological record documents this anomaly clearly. Sites dated between 100,000 and 70,000 years ago show rapidly increasing technological complexity. Heat-treated stone tools at Pinnacle Point, 72,000 years ago. Composite wooden digging sticks with bone points at Border Cave, 80,000 years ago. And symbolic artifacts, appearing with sudden sophistication.
There was a kind of behavioral revolution 50,000 years ago, notes Dr. Richard Klein of Stanford University. Anatomically, modern humans probably evolved in Africa 200,000 years ago, but initially they were behaviorally much like the Neanderthals.
Then around 50,000 years ago, those modern human anatomy and modern human behavior came together, and that led to the dispersal of modern humans to Eurasia. But what prompted this revolution? The genetic foundation for our cognitive abilities must have existed before being expressed in behavior. But what catalyst triggered such dramatic expansion? Some researchers propose a genetic mutation affecting brain function might have spread rapidly. Others suggest existing cognitive capabilities found expression through cultural rather than biological evolution. Yet these explanations struggle to account for both the timing and comprehensiveness of the change. The evidence presents an evolutionary anomaly, a cognitive acceleration without precedent in nature. This unexplained leap raises profound questions about human origins. If standard evolutionary pressures can't fully explain this cognitive explosion, what catalyst triggered such a dramatic change? This question continues to challenge our understanding of human evolution and opens the door to explanations beyond conventional scientific models. Brain architecture anomalies. The human brain represents one of the most complex structures in the known universe, containing approximately 86 billion neurons forming trillions of connections. While all primate brains share basic architectural features, the human brain contains specialized structures and neural networks that set it apart in ways that challenge conventional evolutionary explanations. Over the course of human evolution, our brain size has increased dramatically. Early Australopithecines had brains averaging 400 to 500 cubic centimeters, similar to modern chimpanzees. By the time Homo erectus appeared around 1.8 million years ago, brain volume had increased to 800 to 1,100 cubic centimeters. Modern humans possess brains averaging 1,300 to 1,500 cubic centimeters, roughly triple the size of our earliest ancestors. This represents an extraordinary rate of growth compared to other evolutionary changes. However, size increase alone doesn't explain human cognitive uniqueness. The human brain differs from other primate brains in specific structural and functional adaptations that have no clear evolutionary precedent. These differences appear primarily in regions associated with language processing, abstract thinking, and social cognition. Precisely the capabilities that most dramatically separate humans from other species. The cerebral cortex, the wrinkled outer layer responsible for our highest cognitive functions, makes up approximately 80% of the human brain compared to 30 to 40% in most mammals. This expansion concentrates in areas with limited immediate survival value. The prefrontal cortex, responsible for planning, moral reasoning, and impulse control, is proportionally twice as large in humans as in chimpanzees. Like an advanced computer network that suddenly received specialized processors rather than gradual upgrades, two brain regions highlight this evolutionary mystery: Wernicke's area and Broca's area. These interconnected regions form our language processing system, with Wernicke's handling comprehension and Broca's managing production. While chimpanzees possess precursor regions, they lack the neural architecture for processing grammar and syntax. The specialized circuitry for language appeared with remarkable speed rather than developing gradually. The mirror neuron system presents another evolutionary puzzle. These neurons activate both when performing an action and when observing others perform the same action. In humans uniquely, this system extends beyond simple action recognition to enable complex social cognition, including theory of mind, understanding that others have different thoughts and beliefs. This integrated network connects to language centers and emotional regions in ways not seen in other primates,
creating the foundation for empathy and cultural transmission.
Similarly, the human striatum, involved in processing reward and motivation, shows distinctive development patterns.
This structure enables the complex social bonds forming human societies, with neural pathways significantly different from other primates. In essence, multiple brain systems reorganized simultaneously to create our unique cognitive abilities. The timeline of these adaptations is particularly striking. Analysis of fossil endocasts shows significant changes in brain organization occurred within a compressed time frame.
The parietal lobes, involved in spatial cognition, and cerebellum, crucial for coordinating complex movements, show particularly rapid development between approximately 100,000 and 35,000 years ago. This time frame creates a significant problem for evolutionary theory. Conventional natural selection typically requires hundreds of thousands to millions of years to produce complex adaptations. The neural architecture supporting human cognition appears to have developed far more rapidly than standard models predict. As Dr. Terence Deacon of UC Berkeley notes, the human brain has been substantially reorganized. The rapid development of specific neural circuits devoted to language and social cognition represents an evolutionary anomaly that we still can't fully explain. This reorganization includes pronounced brain asymmetry, with the left hemisphere specialized for language and the right for spatial processing. The arcuate fasciculus, connecting Wernicke's and Broca's areas, is significantly larger and more complex in humans than other primates, with no clear evolutionary precursors. Some researchers propose that minimal genetic mutations triggered cascading effects in brain development.
The FOXP2 gene differs between humans and chimpanzees by just 2 amino acids, yet influences the development of neural circuits critical for language.
However, these genetic explanations struggle to account for the speed and specificity of neural adaptation. Others suggest gene culture co-evolution, where cultural innovations create selective pressures driving genetic changes.
While this helps explain continued cognitive development, it doesn't fully address the initial emergence of neural architecture necessary for complex culture.
The human brain's unique features raise a compelling question. Could such specific and complex structures have developed so rapidly through conventional evolutionary processes?
The unprecedented speed and precision of these adaptations suggest either unknown mechanisms of neural evolution or fundamental gaps in our understanding of human cognitive development. As neuroimaging technology advances and our understanding of the genetic basis of brain development improves, we may eventually unravel this mystery. Until then, the unique architecture of the human brain remains one of the most profound puzzles in our evolutionary story.
The genetic programming mystery.
Within the vast stretches of our non-coding DNA lies a mystery that challenges our understanding of human genetics. Scientists have discovered intricate systems that regulate gene expression with precision that resembles sophisticated computer programming more than random evolutionary developments. These regulatory elements don't create proteins themselves but instead control when, where, and how our protein coding genes activate, functioning as a complex operating system for our cellular machinery.
For decades, scientists focused almost exclusively on the 2% of our genome that codes for proteins. However, research now reveals that non-coding regions contain regulatory elements that act as genetic switches, turning genes on and off in highly specific patterns. Like the onboard software of a finely tuned machine, these switches respond to cellular conditions, developmental stages, and environmental signals, creating a dynamic system of genetic regulation that determines everything from basic cellular function to complex cognitive processes. Dr. Jennifer Doudna, molecular biologist at UC Berkeley, explains, what we're discovering in non-coding DNA is highly organized regulatory systems.
These sequences control the precise timing and location of gene expression in ways that are essential for proper development and function.
The complexity of these regulatory mechanisms exceeds what scientists initially expected.
Many non-coding elements function like algorithmic programs, processing multiple inputs and producing coordinated outputs that regulate entire networks of genes. These systems involve feedback loops, redundancy checks, and conditional logic that bears striking resemblance to human-engineered computer code.
The ENCODE project revealed that approximately 80% of our genome performs some biochemical function, primarily related to regulating gene expression.
This finding contradicted the view that non-coding DNA was mostly junk left over from evolution.
Instead, this genetic material contains essential instructions for cellular operation and development.
Research from multiple institutions has demonstrated that many diseases result from changes in non-protein coding sequences.
Studies at the Broad Institute identified thousands of mutations in regulatory regions associated with conditions ranging from autism to heart disease, highlighting the crucial role these sequences play in proper cellular function. These regulatory systems ensure genes activate at exactly the right time, in precisely the right amounts, and only in specific cells or tissues.
During brain development, for example, regulatory elements coordinate the expression of thousands of genes in a precise sequence that builds the neural architecture supporting human cognition.
Dr. Manuel Aries, RNA researcher at UC Santa Cruz, notes, "The precision with which these regulatory networks operate is extraordinary. They maintain homeostasis while allowing for appropriate responses to changing conditions.
It's a level of complexity that we're still working to understand fully." Among the most mysterious components are long non-coding RNAs, LNC-RNAs. The SRA1 gene, for example, produces both a protein-coding transcript and a non-coding RNA that regulates hormone receptors, demonstrating how these elements can serve multiple purposes within our genetic programming. The human genome contains thousands of LNC-RNAs, many involved in brain development and cognitive function.
These LNC-RNAs control entire gene networks.
The XIST-LNC-RNA coordinates the inactivation of an entire X chromosome in
female cells, a process involving hundreds of genes.
Other LNC-RNAs like HOTAIR regulate developmental gene expression across multiple chromosomes, ensuring proper body patterning during embryonic development. What puzzles scientists is how these complex regulatory systems evolve. According to conventional evolutionary theory, complex systems develop gradually through incremental improvements. Yet some regulatory elements in the human genome appear to have emerged suddenly, without the expected precursors in our evolutionary relatives. Comparative genomic studies reveal that certain regulatory elements controlling cognitive development appear uniquely in humans or show significant differences from their counterparts in other primates. These elements often regulate genes involved in brain development and cognitive functions, precisely the characteristics that most dramatically distinguish humans from other species. The FOXP2 gene provides a striking example. Often called the language gene, FOXP2 plays a crucial role in speech and language development. While the FOXP2 protein is highly conserved across mammals, the regulatory regions controlling its expression show human-specific changes that appeared with suspicious suddenness. These regulatory differences create unique patterns of FOXP2 expression in the human brain, particularly in regions associated with language processing.
Dr. Catherine Pollard, computational biologist at the Gladstone Institutes, explains, "When we look at the regulatory regions around genes involved in brain development, we see human-specific changes that emerged relatively recently in evolutionary terms. Some of these changes appear to have occurred without the gradual progression we would expect from standard evolutionary processes. The sophistication of these regulatory systems raises profound questions about their origins.
Their computer-like precision, their apparent sudden emergence in the human genome, and their specific focus on cognitive functions challenge conventional explanations of how such complex systems evolve." As Dr. Eugene Koonin, evolutionary biologist at the National Center for Biotechnology Information, notes, "The complexity of genetic regulatory networks presents a significant challenge to evolutionary theory.
How such elaborate control systems evolve remains one of the major unanswered questions in biology. For over a century, paleontologists have combed the earth searching for fossil evidence to illuminate our origins.
This search has yielded remarkable discoveries, from Lucy, the famous Australopithecus, to the recently discovered Homo naledi. Yet despite these findings, a significant problem persists. When scientists examine the fossil record for evidence explaining our extraordinary cognitive development, they encounter a puzzling absence of transitional forms. According to established evolutionary principles, the fossil record should contain a clear progression of specimens showing gradual, incremental increases in brain size and skull morphology. We would expect to find a series of intermediate forms documenting the steady expansion of the brain case and the development of anatomical features associated with advanced cognition. These transitional fossils would chart the path from primitive hominids to fully modern humans with their unprecedented mental capabilities. Instead, what we find is a significant gap in the fossil record precisely when human cognition was supposedly making its most dramatic leap forward. This gap resembles missing chapters in humanity's autobiography, crucial pages torn out exactly where the most important part of our story should be told.
The timeline of brain evolution reveals this problem clearly. Early hominin brain size increased only slightly from 6 to 2 million years ago, with early Australopithecus species having brains around 400 to 500 cubic centimeters, comparable to modern chimpanzees. From 2 million to approximately 800,000 years ago, brain size increased somewhat more rapidly, but primarily in proportion to increases in overall body size. The critical period occurs between approximately 800,000 and 200,000 years ago. During this time, brain volume grew significantly, approaching the modern human average. This acceleration coincides with multiple glacial and interglacial cycles, creating environmental stress. However, environmental pressures alone don't adequately account for this rapid growth. Other mammals facing identical climate changes didn't develop comparable cognitive adaptations. The relationship between brain size and intelligence presents another puzzle. While brain volume approached modern levels around 300,000 years ago, some modern humans with brain sizes similar to Homo erectus display entirely normal intelligence. Conversely, Neanderthals had slightly larger brains than modern humans on average, yet didn't achieve the same level of technological innovation or symbolic expression. This suggests that neural architecture matters more than simple size. Yet the fossil record preserves little direct evidence of these crucial internal structures, creating another gap in our understanding, like having the outside of a computer but no circuit boards to explain how it functions. Dr. Tetsuro Matsuzawa's cognitive trade-off hypothesis suggests humans sacrifice certain immediate memory capabilities found in other primates to gain language and higher cognitive functions. While this explains some cognitive differences between humans and apes, it fails to account for the timing and emergence of our expanded abilities. The most perplexing aspect emerges when examining the archaeological timeline. Anatomically, modern humans appeared around 300,000 years ago, yet evidence for behaviors reflecting modern cognition, symbolic art, complex tools, ritual practices, appears consistently only about 100,000 years ago, becoming widespread around 50,000 to 40,000 years ago. This creates a 200,000-year gap between anatomically modern brains and evidence of modern thought. If the physical brain capable of advanced cognition existed for hundreds of thousands of years before those abilities were expressed, what triggered their eventual emergence? The fossil record provides no clear answer to this crucial question. As Dr. Ian Tattersall of the American Museum of Natural History notes, there's a huge and very puzzling disconnect between the archaeological record of Homo sapiens behaviors and the record of anatomical Homo sapiens origins. The appearance of the anatomical entity preceded the appearance of the behaviors that we associate with it by a huge amount. Various hypotheses attempt to explain this gap, from preservation biases to the possibility that cognitive changes resulted from small genetic modifications not reflected in skeletal anatomy. Some propose that the capacity for advanced cognition evolved earlier but remained unexpressed until cultural factors triggered its manifestation. Others suggest neural reorganization rather than brain size might explain cognitive advancement. These explanations, while plausible, leave significant questions unanswered. Despite extensive searching, the fossil record has not yielded the progressive series of transitional forms that would document the gradual development of human cognitive capabilities. Instead, it presents a pattern of physical evolution followed by an apparent cognitive revolution that occurred with unexpected suddenness. This absence of transitional forms forces us to consider alternative possibilities. Might the missing links suggest a development not preserved in fossilized bones? Could the cognitive leap have resulted from changes that wouldn't leave direct evidence in the fossil record? Whatever the explanation, the fossil evidence presents more questions than answers about our rapid cognitive evolution. Genetic markers without evolutionary precedent. When we examine the human genome through the lens of evolutionary biology, we expect to find clear genetic lineages. According to established evolutionary principles, new genes and genetic features should develop through gradual modification of existing genetic material. These changes leave traceable evolutionary pathways, allowing scientists to follow the development of specific genes across species and through time. Every significant genetic innovation should have precursors and intermediate forms in our evolutionary relatives, a genetic fossil record paralleling the physical one. Yet when geneticists analyze human DNA, they encounter anomalies that challenge this fundamental principle. Our genome contains sequences that appear to have emerged fully formed, with no clear evolutionary pathway leading to their development. These genetic features show few or no precursors in other primates, appearing suddenly in the human lineage rather than evolving gradually through natural selection.
The srgap2c gene exemplifies this phenomenon. This gene plays a crucial role in human brain development, specifically in forming dendritic spines, small protrusions on neurons that receive signals from other brain cells. The density of these spines directly affects neural connectivity and processing power, with humans having significantly more than other primates. srgap2c emerged approximately 3.4 million years ago through a partial duplication of srgap2. This duplication created a truncated version that inhibits the original gene's function, allowing more complex neural connections to form. While gene duplications occur commonly in evolution, srgap2c appears to have emerged in a single event rather than through gradual modification. Dr. Frank Poyo of Columbia University notes, the emergence of srgap2c represents a single genetic change that may have contributed significantly to human brain evolution. Von Economo neurons, VENs, present another evolutionary puzzle. These specialized brain cells, associated with social awareness and intuitive decision-making, exist in humans, great apes, elephants, and certain cetaceans, suggesting independent evolution across multiple species rather than inheritance from a common ancestor. In humans, these neurons are far more abundant, with their developmental genes showing rapid modifications unique to our lineage. The modified FOXP2 gene, crucial for language development, contains two human-specific amino acid changes that affect its function. These modifications appear to have emerged from spread rapidly within the last 200,000 years, an extremely brief period in evolutionary terms. Mutations in this gene cause severe speech disorders, highlighting its importance in our unique communication abilities. Scientists have identified over 2,700 human accelerated regions, HARs, DNA stretches that remained unchanged in most mammals but suddenly began evolving rapidly in humans. These regions primarily regulate genes involved in brain development. Dr. Catherine Pollard, who identified the first HAR, explains, these regions were highly conserved through hundreds of millions of years of evolution, then suddenly started changing rapidly in the human lineage. Microcephalin and ASPM genes, associated with head size and brain development, show unexpected patterns. When mutated, they cause microcephaly, a condition characterized by a significantly smaller brain. However, population genetic studies reveal these genes show little evidence of being under strong selective pressure in current human populations, contradicting what we'd expect if they drove brain size evolution through natural selection. Perhaps most mysterious are genetic elements that appear to have entered the human genome with no evolutionary precursors whatsoever. Some DNA sequences show patterns suggesting insertion rather than evolution from existing genetic material. These insertions often involve regulatory elements controlling gene expression during brain development and cognitive function. When scientists attempt to trace their evolutionary history using comparative genomics, they find no clear path showing gradual development from ancestral forms. These genetic anomalies resemble sudden software updates rather than the gradual code revisions we'd expect from natural selection. Their appearance challenges conventional understanding, like finding modern components in ancient machinery with no intermediate versions. The conventional explanation involves mechanisms like gene duplication or horizontal gene transfer, followed by rapid selection. Yet the specificity, complexity, and timing of these genetic innovations remain difficult to explain through standard evolutionary processes. As we continue studying these genetic markers without clear evolutionary precedent, we face the possibility that our understanding of human genetic development may be incomplete. The presence of these anomalies represents one of the most significant mysteries in human origins. If these sequences didn't evolve gradually through natural selection as evolutionary theory predicts, how did they appear in our genome? This question stands at the frontier of human genetics, challenging us to reconsider how our species acquired its most distinctive biological features. The ancient intervention hypothesis. Among the ancient civilizations that flourished in Mesopotamia, the Sumerians stand out not only for their remarkable innovations but for the thousands of clay tablets they left behind. These artifacts, dating back approximately 6,000 years, document the world's earliest known writing system, cuneiform script pressed into soft clay and preserved through millennia. The Sumerian tablets record everything from mundane business transactions to elaborate cosmological narratives that describe their understanding of human origins. These ancient texts consistently reference beings called the Anunnaki, described as advanced entities who came to Earth from another world. The wedge-shaped characters,
meticulously pressed into wet clay with reed styluses, created intricate patterns that scholars have spent decades deciphering. Each tablet, hardened by time, carries within its surface detailed accounts that seem to blend mythology with startlingly technical information. The Sumerian creation texts, particularly the Enuma Elish and Atrahasis epic, describe the Anunnaki as beings who arrived on Earth to extract minerals, especially gold, required for their home planet. According to these narratives, the Anunnaki initially performed this mining work themselves but eventually rebelled against the difficult labor. This labor crisis precipitated what the texts describe as a profound intervention in Earth's evolutionary history. In response to this labor shortage, the tablets describe a decision made by the Anunnaki leadership to create a worker species. The texts specifically name Enki, also called Ea, and Ninhursag as the architects of this intervention. What follows in these ancient accounts is a description of what appears to be genetic engineering, the manipulation of existing hominid DNA to create an advanced hybrid worker. The texts refer to this new being as the Adamu, a term bearing obvious similarity to the biblical Adam. According to translations, the Sumerian tablets describe a process where the essence, or blood, of an existing hominid was mixed with the essence of the Anunnaki to create a hybrid being with greater capabilities than the native species but with specific limitations. This alleged genetic intervention occurred approximately 300,000 years ago, aligning with the current scientific consensus about when anatomically modern humans first appeared in the fossil record. Paleontological evidence places the emergence of Homo sapiens in Africa between 300,000 and 200,000 years ago. The tablets contain descriptions of multiple creation attempts, with early versions having various problems requiring correction. These accounts include references to concepts that would have been entirely unknown to ancient people, discussions of genetic traits, inheritance patterns, and descriptions of fertility problems in early hybrid specimens. The texts describe issues with the initial created beings, including an inability to reproduce, which required further genetic modifications. The accounts detail deliberate limitations allegedly programmed into these created humans. The tablets describe specific modifications to reduce workers' lifespan and limit their intelligence, ensuring they would be capable workers but not rivals. These modifications parallel the actual differences between humans and other primates. Shorter lifespans than predicted by body size and dramatically different cognitive abilities from our closest evolutionary relatives. Later tablets describe a final genetic version of humans that incorporated changes to brain capacity. According to translations, certain Anunnaki leaders eventually granted humans additional intellectual capabilities, against the wishes of others who preferred keeping the created workers limited. This narrative of competing agendas regarding human development appears in multiple Sumerian sources. Conventional archaeology interprets these accounts as purely mythological, creative narratives developed by ancient people trying to explain their origins through stories of gods and supernatural forces. From this perspective, the Anunnaki represent deified rulers or natural phenomena, and the creation accounts reflect metaphorical understandings rather than literal descriptions. Yet scholars like Zachariah Sitchin and Samuel Kramer have noted specific features in these texts that challenge conventional dismissal. The Sumerian civilization emerged fully formed around 6,000 years ago with advanced mathematics, astronomy, agriculture, and metallurgy, showing little evidence of gradual development. Their texts display knowledge of the solar system that wasn't rediscovered until the modern era, including descriptions of planets visible only with telescopes. The question becomes more pointed. How and why would ancient Sumerians create accounts describing what appears to be genetic engineering if they had no concept of genetics? The concepts of DNA, genes, and genetic manipulation lay millennia in the future. Even the basic understanding that reproduction involves the combination of genetic material from two parents wasn't established until the 19th century. While mainstream archaeology dismisses these texts as mythology, their specific descriptions of genetic manipulation raise a provocative question. Could these ancient accounts preserve a record of actual events? The parallels between Sumerian creation stories and the unexplained aspects of human evolution remain one of the most controversial intersections of ancient texts and modern genetic science. Competing scientific explanations. Mainstream science offers several competing explanations for the unusual evolutionary trajectory of human intelligence. Each theory approaches the puzzle from a different angle, attempting to explain how natural processes could have produced our remarkable cognitive abilities. These models share a common goal:
to account for human intelligence within the framework of evolutionary theory without invoking external intervention or design. The social brain hypothesis, developed by British anthropologist Robin Dunbar, proposes that human intelligence evolved primarily as an adaptation for navigating increasingly complex social environments. As early human groups grew larger, individuals needed to track relationships, anticipate behaviors, and manage social standing. This theory gained support from the correlation between brain size and social group size across primate species. However, it struggles to explain human uniqueness. Many species like chimpanzees, elephants, and dolphins live in complex social groups, yet none developed human-level intelligence. Bottlenose dolphins, for instance, maintain intricate social networks requiring sophisticated communication, yet their cognitive evolution followed a different path. The ecological dominance social competition model, proposed by Mark Flynn, suggests that as early humans gained ecological advantage through tool use and cooperation, selective pressure shifted from environmental challenges to social competition within the group. Intelligence became valuable as humans competed with each other for status and resources. This created a feedback loop where tool use reduced environmental pressures, shifting competition to within-group dynamics that favored cognitive abilities. While addressing some weaknesses of the social brain hypothesis, this model struggles with timing. Archaeological evidence shows gradual ecological dominance, yet cognitive capabilities appear to have developed rapidly during specific periods. The sexual selection model, advanced by Geoffrey Miller, approaches the question from a reproductive perspective. Intelligence served as a fitness indicator, with early humans preferentially selecting partners displaying greater cognitive abilities. Traits like language, creativity, humor, and storytelling evolved as courtship adaptations, creating a self-reinforcing cycle where cognitive abilities became increasingly elaborate. This helps explain cognitive development beyond survival needs but doesn't adequately address why sexual selection for intelligence occurred specifically in humans and not other species that use elaborate mating displays. Each explanation offers insights into human cognitive evolution, yet none provides a complete picture on its own. The cultural intelligence hypothesis, proposed by Michael Tomasello, suggests that brain size increased to support the cognitive demands of cultural information transmission. As humans accumulated and shared knowledge across generations, larger brains were favored to store and process this information. This theory highlights our unique capacity for cumulative culture, building on previous generations' knowledge rather than starting from scratch. It explains the gradual acceleration of technological advancement but faces a chicken and egg problem. Cultural accumulation requires advanced cognitive abilities to begin with. The reduced aggression theory, associated with Richard Wrangham's work, proposes that decreased aggression enabled advanced intelligence. Humans essentially self-domesticated, with less aggressive individuals gaining social advantages leading to cognitive changes. Physical changes in modern humans parallel those in domesticated animals. Reduced facial robusticity, smaller teeth, decreased sexual dimorphism, and juvenile facial features. Studies of domesticated foxes support this, as they developed enhanced social cognition after being selectively bred for tameness. However, this theory alone doesn't fully explain our capacity for abstract thought and innovation. Scientists increasingly recognize that no single theory fully accounts for human intelligence. These mechanisms likely worked in combination, creating feedback loops that accelerated cognitive development beyond what any single evolutionary pressure could achieve. For example, tool use might have initially evolved for ecological advantage, then became subject to social learning and cultural transmission, while simultaneously affecting mate selection patterns and social structures. The limitations of these explanations reveal a scientific community still searching for a comprehensive understanding. Even when combined, these models struggle to explain the emergence of symbolic thinking, language, and abstract reasoning that characterize modern human cognition. Consider how the earliest cave paintings from 40,000 years ago required not just technical skill but abstract symbolic thinking, a capacity that seems to have appeared relatively suddenly in evolutionary terms. This gap in our scientific understanding highlights the complexity of the evolutionary processes that shaped our species. As research continues in genetics, paleoanthropology, and comparative psychology, our understanding of human cognitive evolution continues to evolve.
incorporating new evidence and theoretical frameworks to address these outstanding questions about our origins.
If advanced beings modified our genetic code, would we be able to detect their technological signature? This question might sound like science fiction, but recent discoveries in genomics have revealed patterns in human DNA that some researchers suggest appear suspiciously engineered rather than naturally evolved. As we've developed more sophisticated tools for analyzing the genome, scientists have uncovered regulatory systems of surprising complexity that control our genetic expression. Detecting engineered DNA might work similar to how digital forensics experts identify watermarks in modified images.
While natural evolution produces systems through accumulated small changes, engineered systems typically display elegant efficiency, redundancy features, and optimization for specific purposes.
Geneticists could theoretically identify such engineering by looking for signatures distinct from patterns created by natural selection, mutation, and genetic drift. Among the most striking examples are bifunctional genomic loci, regions of DNA that serve dual purposes. These remarkable segments express both messenger RNA, mRNA, that codes for proteins, and non-coding RNA that regulates other genes.
This dual functionality allows a single DNA segment to perform multiple functions, with an efficiency resembling technological design principles. These regions function either in mutually exclusive fashion, producing different RNA molecules under different conditions, or simultaneously producing both types of RNA.
The SRA1 gene exemplifies this sophisticated arrangement. It expresses both an oncogenic protein affecting cell proliferation and a long non-coding RNA regulating hormone signaling pathways. What makes this remarkable is its intricate system of
alternative splicing sites that produce either protein-coding mRNA or regulatory non-coding RNA depending on cellular conditions.
The precision of this switching mechanism responds to specific cellular signals with extraordinary sophistication. Similarly, the PPP1R10 gene expresses both a protein-regulating RNA processing and a long non-coding RNA through alternative splicing that breaks the normal protein coding sequence. This arrangement enables function in multiple biological pathways simultaneously, maximizing information content in limited genetic space. The control mechanisms regulating this functionality show a precision that challenges conventional evolutionary explanations. Many of these sophisticated regulatory mechanisms control genes involved in brain development and cognitive function. The precision with which non-coding DNA regulates gene expression in the human brain resembles a control system maintaining specific cognitive capabilities within certain boundaries. These networks ensure neural development follows precise patterns, activating genes in exact sequences to build the brain's complex architecture.
Dr. Eugene Stanley, a computational biologist at Boston University who studies genetic regulatory networks, notes, "The regulatory systems controlling brain development show an unexpected level of organization.
They coordinate the expression of thousands of genes with a precision that's difficult to explain through random processes alone."
The more we understand these systems, the more they resemble designed control networks.
The FOXP2 gene, associated with language development, is controlled by a complex network of regulatory elements ensuring activation in specific brain regions during precise developmental windows. Mutations affecting its regulatory
regions can cause severe language impairments.
This control system demonstrates robust design with multiple regulation layers ensuring proper function even under varying conditions.
Certain regulatory elements in the human genome function as genetic subroutines, modular components performing specific functions that can be activated in different contexts. This modularity resembles software design, with reusable code components called upon when needed. Natural selection typically produces less modular systems unless specific pressures favor modularity.
These observations prompt a provocative question. Could some regulation mechanisms in our genetic code have been artificially designed?
While mainstream science attributes these sophisticated systems to natural evolutionary processes not yet fully understood,
some researchers suggest certain genetic regulatory systems show hallmarks of technological design rather than gradual evolutionary development.
Dr. Robert Lanza, a noted biologist, has commented, "As we decode more of the genome, we're finding levels of information processing that exceed our expectations for naturally evolved systems."
While this doesn't necessarily indicate external design, it does suggest that our understanding of evolutionary mechanisms remains incomplete.
The mainstream explanation involves natural selection, genetic drift, and possibly undiscovered evolutionary mechanisms.
Most biologists suggest that apparent design may reflect our incomplete understanding of how complex regulatory systems evolve through natural processes over millions of years. As genetic research advances, will we find definitive technological signatures in our DNA, or discover new evolutionary mechanisms explaining these sophisticated systems? Whatever we uncover, the extraordinary complexity encoded in our genome continues to challenge our understanding of human origins and may yet reveal surprising truths about our genetic heritage. The evolutionary timing paradox. When we examine human history through archaeological evidence, we encounter a timeline paradox that challenges conventional evolutionary models. For most of human existence, spanning hundreds of thousands of years, technological development progressed at an extraordinarily slow pace. The archaeological record reveals that early stone tools remained remarkably consistent in their design and function for immense periods of time, showing only minor refinements across hundreds of millennia. The Oldowan tool industry, characterized by simple stone choppers and flakes, emerged around 2.6 million years ago and persisted with minimal change for nearly a million years. The Acheulean tradition, recognizable by its symmetrical hand axes, appeared approximately 1.7 million years ago and remained dominant for over a million years. This consistency suggests the cognitive capabilities of their creators remained relatively stable across countless generations. This pattern of technological stasis presents a significant puzzle. If the human brain evolved gradually, we would expect to see a corresponding improvement in technological sophistication. Yet the archaeological record shows long periods of minimal innovation followed by sudden accelerations that cannot be explained by gradual genetic changes. Imagine a world where technology barely changes for hundreds of thousands of years. Then suddenly, around 50,000 years ago, human innovation accelerates dramatically. This period, known as the Upper Paleolithic Revolution or Creative Explosion, transformed our understanding of cognitive evolution. Tools that had remained virtually unchanged were rapidly replaced by sophisticated implements designed for specific purposes. Blade technologies, bone tools, fishing equipment, and advanced hunting weapons appeared with remarkable suddenness. This acceleration intensified around 30,000 years ago, when the archaeological record shows widespread emergence of fully modern behavior patterns. Figurative art, musical instruments, complex tools, personal ornamentation, and evidence of ritual practices appear across continents with a suddenness that archaeologists struggle to explain through conventional evolutionary models. The cave paintings at Chauvet Cave in France, dated to approximately 32,000 years ago, exemplify this sudden sophistication. These artworks display advanced techniques including perspective, shading, and anatomical accuracy that suggest a fully developed artistic tradition without clear developmental precursors. Archaeologist Jean Klotz noted, "The artists of Chauvet were as technically accomplished as any that followed them. Their work represents a profound and sophisticated aesthetic sensibility that appears fully formed in the archaeological record." Similarly, Venus figurines, small sculptures depicting female forms, appeared across Europe between 35,000 and 25,000 years ago with consistent stylistic elements despite their wide geographical distribution. The bone flutes discovered at Geisenklosterle Cave in Germany, dated to approximately 36,000 years ago, feature evenly spaced finger holes capable of producing specific musical scales, indicating both technical skill and theoretical understanding of musical intervals. The dramatic turning point in this timeline creates a profound paradox. Anatomically modern humans had already existed for over 150,000 years before this creative explosion occurred. Fossil evidence indicates humans with essentially modern skeletal anatomy appeared in Africa around 200,000 years ago. Yet the archaeological evidence for modern behavioral patterns didn't consistently appear until much later, creating a gap of at least 100,000 years. Paleoanthropologist Ian Tattersall addresses this disconnect. There's a huge disconnect in timing between when we became the anatomical creatures we are today, and when we started behaving in a symbolic manner. That's a gap of at least 100,000 years that's very difficult to explain. This pattern suggests certain cognitive capabilities may have been activated rather than evolved, that the genetic potential for advanced cognition existed long before being fully expressed in human behavior. Modern human cognitive capabilities may have emerged rapidly when specific neural systems reached completion or were triggered by environmental or cultural factors. The evolutionary timing paradox becomes even more striking when examining specific innovations. Sewing needles around 30,000 years ago enabled tailored clothing,
significantly improving survival in cold environments. Advanced projectile weapons like the Atlatl dramatically increased hunting efficiency. These innovations appeared suddenly rather than through gradual refinement. This acceleration in innovation occurs too rapidly to be explained by genetic changes alone, which typically require thousands of years to spread through populations. The flourishing of technological innovation observed in the archaeological record happens faster than genetic evolution would allow. The paradox raises fundamental questions about human cognitive evolution. If the genetic potential for advanced cognition existed in humans for over 100,000 years before being consistently expressed, what triggered its eventual emergence? Why did human creative and technological development remain relatively static for thousands of generations, then suddenly accelerate within a few millennia? This explosion of human creativity forces us to consider alternative explanations. Was this sudden flourishing the result of a genetic program activating at a specific developmental point? Did cultural factors reach a critical threshold triggering innovation? Whatever the explanation, the timing paradox in human technological development represents one of the most significant puzzles in our evolutionary history. The plasticity puzzle. The human brain possesses a remarkable ability to reorganize itself throughout life. This capacity for neural plasticity, the brain's ability to form new connections and adapt to new information, represents one of the most distinctive features of human biology. While many species exhibit some degree of neural plasticity, humans display this trait at levels that far exceed those of any other organism on Earth, including our closest primate relatives. Neural plasticity occurs when the brain creates new synaptic connections or modifies existing ones in response to experience, learning, or injury. Most animals experience plasticity primarily during early development, with brain malleability decreasing as they mature. Cats require proper visual input during a critical period to develop normal sight, after which their visual cortex becomes significantly less adaptable. Humans break this pattern completely. Our brains maintain extraordinary plasticity throughout our entire lives. This exceptional flexibility enables us to learn new languages in adulthood, master complex skills decades into our lives, and recover function after brain injuries. Consider stroke patients who relearn speech through therapy as their brains form new neural pathways to bypass damaged areas, a testament to our remarkable neural adaptability. The differences between human and animal plasticity are stark. Humans, lifelong learning capacity with minimal decline. Primates, significant learning ability that diminishes substantially with age. Other mammals, critical periods after which plasticity sharply decreases. Birds, reptiles, limited plasticity primarily confined to developmental windows. This lifelong plasticity appears specifically programmed into our genetic code through regulatory elements that control how our neural architecture develops and adapts over time. The regulatory mechanisms controlling human neural plasticity show unusual characteristics that some researchers suggest resemble engineered systems more than products of random mutation and selection. Dr. Marcus Pembry, professor of genetics at University College London, notes, the genetic mechanisms controlling human neural plasticity show a level of organization that's difficult to explain through conventional evolutionary processes. These systems maintain developmental stability while enabling extraordinary adaptability. The plasticity-led evolution model proposes that phenotypic plasticity, the ability to produce different physical characteristics in response to environmental conditions, plays a crucial role in evolution by accelerating evolutionary change. North American spadefoot toads illustrate this concept, with tadpoles developing as either omnivores or carnivores depending on their environment, allowing survival in varied conditions. Human neural plasticity, however, operates at an entirely different level. Our brains contain specific regulatory elements within non-coding DNA that control plasticity across multiple scales, from individual synapses to entire neural networks. This sophisticated control system enables continuous neural reorganization throughout life, creating new pathways in response to experience and learning. These mechanisms allow humans to acquire skills, languages, and knowledge far beyond what other species achieve, despite having similar neural building blocks. Chimpanzees share approximately 98.8% of our DNA yet display dramatically reduced neural plasticity. While they learn impressive skills, their capacity for continuous adaptation remains limited compared to humans. Language acquisition clearly demonstrates this difference. Children naturally acquire whatever language they're exposed to, mastering complex grammatical structures without explicit instruction. Though this ability decreases with age, adults can still learn new languages throughout life. Chimpanzees, despite intensive training, never develop comparable linguistic abilities due to less flexible neural pathways. Human neural plasticity appears front-loaded in our genome, programmed to activate specific cognitive capabilities at certain developmental stages. These regulatory systems ensure that certain neural networks form during critical periods while allowing others to remain adaptable throughout life. Language networks develop during specific windows in childhood but maintain significant plasticity into adulthood, maximizing learning potential. Research comparing gene expression in human and chimpanzee brains reveals significant differences in genes controlling synaptic plasticity, particularly in the prefrontal cortex. Many of these differences involve human accelerated regions, HARs, DNA stretches that remained largely unchanged in most mammals but evolved rapidly in humans. A disproportionate number of these HARs regulate genes involved in brain development and neural plasticity. This extraordinary neural plasticity raises profound questions about human cognitive evolution. Was the human brain specifically designed to be a learning machine? The genetic programming enabling our continuous cognitive development appears remarkably sophisticated, with regulatory systems maintaining a precise balance between stability and adaptability throughout life. The significance becomes clearer when we consider what this plasticity enables. Humans build cumulative culture, each generation learning from and building upon previous knowledge. This requires specialized neural adaptations supporting acquisition and transmission of complex information across generations. Whether these genetic mechanisms emerge through conventional evolution or other means remains one of the most fascinating questions in human origins research. What's clear is that the regulatory elements controlling human neural plasticity represented defining features separating us from all other species, enabling our unique capacity for continuous learning, the foundation of human culture, technology, and civilization. As we've explored the mysteries within our DNA, we face a profound choice: accept conventional evolutionary explanations despite their significant gaps, or consider the possibility that our genetic code contains evidence of intervention we're only beginning to detect. The anomalies we've examined raise questions that current science struggles to answer completely. Our remarkable cognitive abilities and complex genetic architecture point to a deeper story. Will you continue to marvel at the secrets hidden in your own cells? Perhaps our greatest mystery lies not in distant stars, but within us, written in a genetic language we're still deciphering, with chapters of human development waiting to be understood.